Hello everyone, it's Dr. Wolfe here, and in this video I'm going to be introducing protein synthesis, aka gene expression. So these two terms mean the exact same thing, all right? And the term synthesis is just a fancy way to say make. So when a gene is being expressed, when it's on, when it's activated, we're really just doing the process of making a protein that DNA codes for. So again, when a gene is on or activated, that means the gene is being expressed. Or in other words, when the gene is on or activated, when we draw it green in our gene expression model, the cell is reading that gene to make a protein. All right, so our goal here is to talk about how is that protein actually made? How do you go from DNA to a protein? So we're starting off with DNA. We've learned as far as that it codes for a protein, specifically the protein structure. And we've learned from our liver lab that that structure is so important for how that protein functions, and then how the protein functions determine how the cells function, which determines how you function, how you are you. All right, and we even learned from the liver lab that if you mess up that structure, right, we heated up our liver, it just kind of deformed it, right, that protein was no longer able to function. All right, so again though, how do we get from DNA to a protein? And notice there's this middle step that we haven't talked about just yet. All right, we can't just actually go from DNA straight to a protein. Instead, there's this middle molecule that needs to be made in order for this process to occur, and it's called RNA. So why can't we just go from DNA to protein? Why do we need this middle step? All right, it has to do with location, location, location. So what I'd like you to do is pause the video, 20 seconds, and on your packet, try to label, predict, what are these structures in the cell? And so now that you've unpaused it, all right, we got our DNA right here, this kind of stringy spaghetti looking stuff. Specifically, it's chromatin because it's in the form where it can be read in that spaghetti form. All right, and DNA is stored in the nucleus. All right, D right here is going to be our cytoplasm, a little bit of throwback to semester 1. It's just the fluid in the cell. And C we haven't talked about yet this year, but this might be a little bit of a throwback to middle school. These are going to be called ribosomes. So these kind of watermelon-looking seeds are going to be our ribosomes. And what do ribosomes do? They make proteins. All right, their job is to actually assemble the proteins. All right, but there's a problem. The instructions for making proteins are located here in the nucleus, aka the DNA is in the nucleus. All right, but the ribosomes that actually make the proteins are out here, and the ribosomes aren't going to make a protein unless they get that message. All right, so this is where RNA comes in. And again, what is RNA? All right, so here we have an RNA molecule, we have a DNA molecule, all right? They're both nucleic acids, so you got carbs, lipids, proteins, and then nucleic acids. All right, notice that DNA is double-stranded while RNA is single-stranded, all right? And both, because they're nucleic acids, are made out of nucleotides, so just a bunch of nucleotides together make a whole DNA molecule or whole RNA molecule. And then on those nucleotides, you can have different letters, right, that are like the DNA code, the RNA code. If we look at DNA, all right, C, G, A, T. We've talked about these letters. And look at it, RNA, C, G, A. Oh, this is different. We have a U. So that's another difference with RNA. Instead of having a T, there's a U. All right, it actually plays the exact same role as what a T would. It's just not a T, it's a U. All right. So how does this process of protein synthesis work? All right, we're actually going to just look at the first stage in this video. I'm going to speed it up. All right, I wanted to give you a little bit of a visual but before we get to like the last section of our notes. All right, so here is our cell, and we're going to zoom into our cell. I got our mitochondria green, zooming into the nucleus, this purple thing right here, and here's our chromosome, right? All this is it's just tightly packed DNA. That's all it is. All right, and then we're going to get to our gene. This could be like the actin gene, so we're going to read it to make an actin protein, muscle fibers. And right here is another protein. Right? Remember, proteins get stuff done in the cell. It's going to read this DNA molecule. Specifically, it's going to open it up, all right, and it's going to essentially kind of make a copy. All right. Now, look at this copy that's being made. Notice that the C goes with the G goes with C, C with G, A with T, all right. But then also notice this U is going with an A. So this strand that's being made right here, this isn't DNA. This is RNA. So we're going from that step of going from a DNA molecule to copying that message on an RNA molecule, all right? So the RNA is being copied, and boom, we got our RNA molecule, all right? So why do we do this, all right? Well, the reason is that RNA, and this is still our RNA molecule, all right? Well, DNA needs to stay in the nucleus. It holds all the codes to make all the proteins, like we need to keep that safe, all right? And if we make a little temporary copy of it in an mRNA molecule, we could just take that message, bring it out to the ribosomes while keeping the DNA message safe. So that's exactly what's going to happen. This mRNA molecule is going to leave the nucleus, and it's going to go out into the cytoplasm, so that's where we are now, and it's going to go to a ribosome, all right? This ribosome is now going to take that code, and it's going to be able to make a protein, all right? Now, there's a little bit more detail in terms of how that ribosome's doing it, but for now, all right, I just want to talk about kind of the big picture and then zoom in on what we just talked about, or the part of the video we just watched. So again, protein synthesis, gene expression, it's all about going from DNA to make a protein, but there's that middle step of going to RNA. That's what we just watched in the video. That's called transcription. Why is it called transcription? Well, back in the day, before printing presses, if you wanted to make a copy of, let's say, the Bible, you had another book that was blank, and you literally just wrote word for word what was going on in this other book. That's how you made the copy. And that's kind of what we saw in the video, right? The mRNA molecule was kind of just this copy of the DNA code, all right? Now, once that DNA code is copied on an RNA molecule, it goes on to the ribosome to make a protein, but you have this DNA code, and now it needs to be translated in order to make a protein. So the two steps of gene expression, protein synthesis, first is transcription, make an mRNA molecule, then translate that message to make a protein. And this GIF, however you pronounce it, shows how this goes down. So we're going to start off with DNA, transcribe to make RNA, translate it to make a protein. And that's really